Okay, about 50. Okay, good afternoon, everyone. I'm Merhaba and hello and everything. And it's nice to see you again. And I hope you had a good week since the last week. So what we're going to do, we've got quite a bit to cover today. And I was saying to the last class that, we'll do our best to follow this material today. And then hopefully the slides are clear. You can go back over and you can make notes and see about the detail we're talking about. Last week we covered the history of biotechnology, and that was quite an easy thing to talk about. Today we're going to be looking at the concept of gene expression and gene regulation. So it's a bit more about the molecular biology of the cell. So we're going to be looking at how DNA goes on to make messenger RNA and how that can be regulated in the cell to save energy. I'll go through this very quickly because we talked about this last week. This is revision. We know that DNA, we know what DNA does in the cell. You've done this before. It has to be replicated, but it also provides the code to make proteins in the cell. And that code goes through an intermediate messenger called messenger RNA. And this is the process transcription to make the messenger RNA. And then that's translated into protein. And I suppose that the main thing that I want to talk about today is how does the cell regulate the synthesis of protein? We know that not all proteins are always made in the cell. They're not always made in the cell, but rather these proteins are made only when the cell needs to make them. And that's what we're going to be talking about today, the regulation of this process. And we find that if the cell can regulate transcription, then ultimately it will end up regulating protein synthesis. Okay, so we're going to talk about really mostly regulation of transcription to save it. This basically saves the cell a lot of energy because when it doesn't need to make these proteins, it doesn't have to do the process of transcription and translation. So we're going to be talking about that regulation. And this is again revision for you. We know that the transcription goes through messenger RNA synthesis. Messenger RNA has these little codons which tells the cell where to put the amino acids in protein in the protein synthesis. Okay. One of the early points I want to make is that, okay, we think of bacteria as being quite simple relative to eukaryotic cells. We know that the bacterial DNA is just a single circle like this, whereas in eukaryotic cells they have chromosomes in pairs like this. But what it's turned out is that actually E. coli are relatively complicated creatures. Bacteria make about 4,000 proteins. And if we take an example of a human, just a random example of a human that I chose, humans make 40,000 proteins. Okay, so actually the difference is not that huge. You think maybe we're much more complicated than bacteria, but bacteria makes 4,000 proteins. We make about 40,000 proteins. So there's actually not a huge number of differences in the number of proteins. And what this means is the complexity of being human is actually linked not to the number of proteins necessarily, but actually the regulation of those proteins. How, when they're switched on, they're switched off, how cells interact and regulate protein synthesis is what makes us human. Whereas E. coli, they look a lot simpler, but they actually also make a lot of proteins. So the point of what we're making here is that gene regulation is really important for making complex individuals like humans, like animals, like mammals, and so on. It's all about how genes are regulated. And I'm not really going to talk too much about eukaryotic cells. We're going to focus on prokaryotes and bacteria. And of course, in bacteria, what we're going to be talking about a lot is the process of transcription and translation, and we're going to focus on prokaryotes. But importantly, for the purposes of this lecture, we're going to talk about the regulation of transcription. And And the basic take-home point that we want to talk about at the beginning of this talk is that if you can regulate transcription, then obviously you regulate protein synthesis as well, because these are all linked, okay? And it makes a lot more sense for a cell to regulate transcription because it saves a lot of energy. And again, you would have covered this in biology and in basic biology and biochemistry, that of course protein synthesis is carried out in ribosomes. Here's the messenger RNA. This is the ribosome adding different amino acids to the peptide, making new proteins in the cell. But the point I want to make is that this actually uses a lot of energy in the cell. Every time a ribosome moves up by one codon and adds a new amino acid, it utilizes 1 GTP just to do this. So you don't have to learn the slide or anything, but what it's showing you is these little protein factories in the cell, lots of polyribosomes, lots of ribosomes making lots of protein. This is very energy utilizing in the cell. It uses a lot of energy in the cell. And the overall point I want to make is that if the cell doesn't need this protein, there's absolutely no point in using all this energy to make something that it doesn't need. Okay, so in other words, if we can save all this energy, it's very beneficial to the cell. So these are the main points are in bold. There's no point in a cell making proteins that it doesn't need. Okay, it needs to, in other words, most proteins in the cell, they're only made when they're needed. And the major means of regulating protein synthesis is actually to regulate the amount of the messenger RNA. Okay, and if you can regulate the amount of message, then you're going to stop certain proteins being made. So in other words, if you can regulate this transcription, then translation doesn't happen. And you can save a lot of energy because you're saving energy here and then you're saving energy there. Okay, so I'm saying that this is revision because you will have covered these concepts in basic biology. This is fundamental biochemistry, basic biology of the cell. Okay, but we're going to focus in a bit more closely on this regulation of transcription, because this is what really saves the energy in the cell. How does a cell sense its environment and then regulate gene expression? So there's a couple of different quizzes populating this lecture. I'm not going to ask. You can interact in the chat if you wish, if you want to give the answer. But what is the answer to this question? Based on what I've just said, what is the best way to save most energy in the cell? Is it by regulating transcription, or is it by regulating translation? Okay, which is the best thing to regulate, given that these two things are linked? And the chat says 1, exactly. So well done. That's exactly it. I just popped out of slideshow. Yeah, exactly. So it's 1. If you can regulate transcription, it's at an earlier stage of the process. You save more energy. Okay, so that's the key take-home message from that. So another key slide in this introduction is this one. So maybe make a note of this, that this is a key slide, because this tells you what is all this lecture all about. What is this lecture about? And it is about this overall concept. And what is represented here on the left is this is the bacterial genome. This is the circular genome. And what I've done is I've said that under normal growth conditions, so E. coli likes to grow at 37 degrees. So at 37 degrees, E. coli is happy. It's making all these proteins, and it's making the proteins by making the messenger RNA and then making the protein. So the messenger RNA is the arrow, protein is the star. So under normal growth conditions, E. coli makes all these proteins, and it's very happy. But what if we switch the E. coli to 42 degrees? It doesn't like 42 degrees, and the cell needs to make other proteins in order to survive. So it makes messenger RNA for other proteins, and it makes these other proteins. You can see I've deliberately put these at different regions, just as an example. So in other words, the cell will switch off. You can see it's not making this protein anymore, but it starts to make a different protein to help it survive. Okay, and you can see that under these conditions, the cell is a bit unhappy. But the concept I want to make is that it switches it switches protein expression. It starts to make proteins that help it survive the stress of high temperature. And what this will tell you is that not all proteins are always made in the cell. The cell is not always making all proteins in the cell, but it responds to its environment to make certain proteins. And it does this by regulating messenger RNA synthesis in the cell. The process of transcription is regulated. And this ultimately regulates the proteins that are made by the cell. So that is the regulation of so this is a key slide, really, I think, which really puts out the concept that we're going to be discussing today. Okay. So again, let's very quickly look, because this is not the key focus, but very quickly look at what happens when a cell undergoes transcription, when it's making messenger RNA for a gene. Okay, whoops. Okay. So again, this is a little bit of revision, because I think you would have covered this before, the fact that the RNA polymerase is what makes the messenger RNA in the cell. And the key fact that the RNA polymerase actually binds to what is known as a promoter region. So today we're going to look a bit more detail at the promoter region, because there are little elements here that we're not going to, maybe you haven't seen before. But this is the process of transcription in the cell. Basically, the RNA polymerase binds to a region of the promoter region, and this starts the process of transcription. Okay, and it's actually helped in its binding by binding to the sigma factor. So what happens is actually the sigma factor actually recognizes the binding site. It attracts in the RNA polymerase, and then this allows the RNA polymerase to start making messenger RNA, which is this red or orange line. Sigma factor is released, and then the messenger RNA is made, and everything dissociates at the end, terminates. So maybe I've gone into slightly too much detail here, but basically here's the sigma factor, which is this little, it's represented by this on the previous slides here, you see. But the sigma factor binds to this portion of the promoter, and it allows the RNA polymerase to bind, and it sets it up in order to make all of this messenger RNA. Okay. So I won't read through these notes. These are just a few notes that describe what I've just described. This is just the text that will help you study and describes what I've just described. And the most important sorry for flicking back and forth, but I think the most important point is that the RNA polymerase binds to this promoter region, and this initiates the process of transcription of the gene. Okay, and it starts to make the messenger RNA and then ultimately the protein. So, so by way of definition, proteins, basically, what we've been talking about, and remember that smiley face picture, we said that E. coli likes to grow at 37 degrees, and then if it moves to 42 degrees, it induces some proteins and it switches off others. And so, just by way of definition, we can define the proteins that are switched on and off in the cell as inducible proteins. These are called inducible. And so actually the vast majority of proteins in a cell are inducible. They go up and down depending on the environment of the cell. And this is really what we're going to talk about today. How the cell regulates protein synthesis, really transcription and then translation. Okay, so for inducible proteins. So in other words, the majority of proteins in the cell are inducible. They go up and down depending on the environment. Whereas there are some proteins that are constitutive. They just are always produced by the cell. It doesn't matter what the environment. They're important proteins. There's actually very few of them, but they may be involved in essential processes, okay, in the cell. Some of these would be actually involved in translation and so on. They're involved in core processes in the cell, okay, and these are called constitutive. They're constitutive because they're always expressed in the cell. So I just wanted to define these two and highlight the fact that we're really going to be talking today about inducible genes. So let's get into the real molecular biology of how these are controlled. So how does the cell control transcription in response to the environment? So how does the environment tell the cell what to do? And there are two systems that I want to talk about very simply. One is called a two-components system, or a two-component sensor regulator. An interesting system, which I'm going to start with, and I've just got 2 slides on this. Okay, so the next two slides are just on this two-component sensor regulator. And then we're going to spend the rest of the lecture talking about repression and induction of gene expression. And we'll get to that when we look at the gene systems. Okay, and actually the first system, this two-component system, is really how the cell responds to stress, like maybe an antibiotic in the environment, or the presence of salt in the environment, and so on. Whereas nutrients is a bit more, there's a bit more detail on that. So the first of these that I want to discuss is the two-component sensor regulator system. And I'm really discussing this as sort of a gentle introduction to the concept, because I really like this sort of the system, this concept, because what it comprises is a number of different proteins, 2 different proteins. The first protein is the sensor, so it's a two-component system. The sensor is present here on the outside of the cell. So this is the cell membrane of the cell. And this sensor is reaching out. It's like a hand reaching out of the cell, and the hand is detecting, it's sort of feeling around to detect certain things that will interact. Okay, and these are specific things that will interact with the protein. So this might be salt. Salt has a sensor which can detect the presence of salt, and also there's also compounds that are like salts called carnitine, and the cell has these receptors that will interact with these. Certain cells as well also have receptors for antibiotics, and we'll see this in the next slide, that some vancomycin resistant cells actually have a two-component system which detects vancomycin, the drug. So in the presence of the drug, the cell will bind to the drug, it'll bind to vancomycin, and it will stimulate a specific response. So what happens when the cell encounters salt, or it encounters whatever this is detecting? So when this cell, so let's take the example of vancomycin. When this cell detects vancomycin as the environmental signal, this is vancomycin here, it detects that vancomycin is present. What this does is this phosphorylates the system. In other words, a phosphate group is transferred. This activates this system by the transfer of a phosphate group. Then what happens is the response regulator, the regulator part, this comes along and this binds to the phosphate, and this becomes activated. This becomes phosphorylated. In other words, you get the transfer of the phosphate group to the response regulator. When the response regulator is phosphorylated, it is activated, and it actively promotes the RNA polymerase to bind to the promoter. Okay, a bit like a sigma factor. Okay, so here you have, here you have vancomycin is detected. This is activated, phosphorylated. The phosphate group is passed to the response regulator. This activates the system, which promotes RNA polymerase binding to the promoter. And what this does is it switches on a number of different genes in the system. Okay, so this activates gene expression. It's an activator. Okay, and so this is highlight it here. This is the exact same sort of scenario. This is just a different way of showing you that, if you have in cells which are vancomycin resistant, what they do is they produce this two-component sensor regulator. The sensor detects the presence of vancomycin. As I've just said, it becomes phosphorylated. It phosphorylates the response regulator, and what this does is it switches on the expression of all these genes that are involved in vancomycin resistance. If the drug is not present, then none of this happens. None of these genes are expressed. Nothing is activated. The cell just sits there and it grows. Okay, and so you start to see, well, this is very clever from the cell. It only needs to express these genes when vancomycin is present, and it has a system which tells it to switch on these genes when vancomycin is present. Okay, so when vancomycin is present, these genes are all switched on, and what they do is these actually make new cell wall components, and they actually make the cell resistant to vancomycin. But it's an energy expensive process, so the cell doesn't need to switch these on all the time. It only needs to switch them on when vancomycin is present. Okay, so that's a very clever system for detecting the stimulant, vancomycin, switching on these genes only when they're needed. So here it acts as an activator of vancomycin resistance. Okay, so I really like this as an introduction to the concept. It's a simple transfer of information. Bang, this is then transferred here. This is transferred to there. This transfer of information triggers gene expression. And in the example of vancomycin resistance, it switches on vancomycin resistance in the cell. Okay, so hopefully when you go back over these slides, you'll be able to see that progression of different concepts. So what about the second scenario? What about the second system that's in place in cells? And this is the key one. This is the key one. And that's why we're going to spend the next 25 minutes talking about this. This is the key system that's in place in cells. Similar systems exist in eukaryotic cells. And this is fundamental biology. And actually, the people who worked out this system in the 1950s in the Pasteur Institute in France, they were awarded the Nobel Prize for this discovery. So Monod and co-workers. So all microbiology students tend to learn this because it's early in the history of microbiology, the history of biochemistry, these concepts. So the first concept here is the fact that in bacteria, genes are very often arranged as operons, and this is the operon concept. And what this means is that the cell has put 3 different genes in this example under the influence of one regulatory unit. So in other words, when RNA polymerase is free, it binds to the promoter. It will express the messenger RNA for 3 different genes, and these are then translated into the 3 different proteins. So it means that these genes are linked because they're all involved in lactose metabolism. So there's no point in having one switched on. You need all 3 of them to metabolize lactose, and so they can be under the same regulatory element. So in an operon, the messenger RNA produced is one long messenger RNA that encodes more than one protein. So here you've got 3 proteins in this example, and an operon has regulatory sites that permit this regulation, which we'll look at now in a moment. Okay, so previously I told you about the promoter, but there's a couple of other sites, the activator site and the operator site, which are also important for what we want to talk about today. Okay, so this is a nice animated slide, which I drew up to try and simplify the process a bit and to describe these different regulatory elements in the cell. Okay, so here you have the LAC operon, the lactose operon. All of these genes encode proteins that are involved in lactose metabolism. But you'll see that the promoter is here. This is where RNA polymerase binds. But there are also 2 other elements. And one is the operator element, And this is where a repressor can bind. So if a repressor binds here, you can see it physically prevents the RNA polymerase from accessing these genes. So it blocks RNA polymerase, and that's why it's a repressor. A repressor will repress gene expression. Here you've got the concept of an activator protein. In this example, the activator binds to an upstream site, the activator site, and this actually promotes transcription. And what this does is it actually helps the RNA polymerase to find the promoter. A bit like a sigma factor, the RNA polymerase interacts with the activator, and this improves gene expression. Okay, so you have two different sites: a site where a repressor can bind to repress, stop, or a site where an activator can bind to activate and improve gene expression from that site. And this is the operon concept, and these sites can control not one gene, but in some cases 5 or 6 or 7 genes all under the same control mechanism. Okay, that was an operon as well we saw in the vancomycin system. That was a number of different genes all regulated. So let's go into the actual scenarios. Let's go into what is actually happening in the cell. And as I said, most of these systems are controlled. In E. coli, they were worked out by looking at metabolism in the cell. So the utilization of sugars or the synthesis of different amino acids and so on. Okay, so we're going to look at one very simple scenario at the beginning, which is what happens in E. coli if the amino acid arginine is present at high levels. Okay, so what you're looking at here is a very simplified version of the pathway for the synthesis of arginine. So in other words, when the cell wants to make the amino acid arginine, wants to make lots of arginine, what it does is it has a synthetic pathway from glutamate all the way through these different steps. So each of these steps is an enzyme. These enzymes make arginine from glutamate, from glutamic acid. Okay. So in this scenario, when arginine builds up in the cell, it becomes a high level. So when the cell has lots of arginine, it no longer needs to make all these. Okay. So in other words, when arginine builds up, the cell doesn't need to make all of these enzymes. And actually what you'll find that it does is it downregulates transcription of the genes. It stops transcription of the genes that are involved in these making of these enzymes. This means no more messenger RNA, no more protein, and this is the regulation of arginine by synthetic enzymes. And what actually happens in the cell is this, that there exists in the cell an arginine repressor here. And this repressor is actually activated by the binding of the co-receptor, which is arginine. Okay, so let's go through these scenarios. At the top, the proteins for arginine biosynthesis. So these are the proteins that make arginine in the cell. They are only made when arginine is absent. So this is what happens. When there's no arginine in the cell, RNA polymerase binds to the promoter, and it transcribes these genes, transcription of gene 1, 2, and 3, and then these enzymes are made. And these enzymes, they actually go on to make arginine, okay, the biosynthetic pathway that we just talked about. But what happens if arginine builds up to a high level in the cell, or if there's lots of arginine in the environment, or there's lots of arginine in the cell? Well, in this case, arginine actually binds to a specific part of the repressor. So arginine binds to this protein. You can see there's a slight change in the shape of the protein when arginine binds to it. You can see that. And now this shaped protein can actually bind to the operator system. So now this can bind. And actually that physically prevents RNA polymerase from expressing these genes. So transcription is blocked. So again, a very clever system, which I'll describe now in the next slide. It's a very clever system whereby if arginine really is at high levels in the cell, that it will block transcription of the genes. Okay, and this is outlined here. What happens when a cell has lots of arginine, arginine all over the place? No enzymes, none of these enzymes are going to be needed. Cell doesn't need to make these, doesn't need to make any more arginine. So what it does is it blocks transcription and it blocks RNA synthesis, and the mechanism is there. This is how it does it. Okay, so it represses gene expression by this route, and it makes sense. If you go back over this later, you'll see that it makes sense. Okay, so another little quiz question, checking the time. So feel free to answer this in the chat if you wish. When the cell has lots of arginine, what does it do? So it's sitting in a pool of arginine. It's got lots of arginine. What does it do? Does it activate the expression of arginine biosynthetic genes to make more arginine? Is it going to make more arginine? Or is it going to repress the expression of arginine biosynthetic genes to make less arginine? Okay, so which do you think it is? Is it 1 or 2? There's quite a few in the chat. And of course, it is 2. Well done. So you all got it right. Well done. So exactly, it represses the expression of the arginine biosynthetic genes. Okay, so these are the sort of concept that the reason I put these little quizzes in, so that when you're studying, you can look at these and say, all right, that's what's happening in the cell. That's why it's happening, and that's how it's happening. So thank you for answering that. So I'm going to actually spend the rest of the lecture talking about lactose metabolism. So we've introduced the concepts there about gene regulation. We've introduced some of the mechanisms. I'm going to go into a bit more detail, you'd be pleased to hear, about lactose metabolism. And the reason I do this is because when the scientists were working out how genes are regulated in cells, they look specifically at lactose, the lac operon. Okay, so now every microbiology student always has to learn about the lac operon, because it was the key part in the history of microbiology, which told us how these systems work. So what happens in the lactose operon? What is actually happening? Oops, I'll just go back. There's something just at the top of my screen, which I will, oops, click off. So lactose, what is lactose? So lactose is a sugar, which is taken up by E. coli by a specific permease system, and then it can be utilized as an energy source. So it's a sugar. It's a sugar which is utilized as an energy source by E. coli in the cell. It's actually a disaccharide, so it's a galactose and a glucose joined together. And not only does E. coli, can it produce this permease to bring in lactose into the cell, but also it breaks apart these sugar molecules to break apart and make the glucose and galactose available. And this is a beta-galactosidase enzyme. So in other words, when lactose is present, the cell needs to make more of these enzymes. Okay, it needs these enzymes and these permeases. So lactose is present, it needs these systems in order to use the lactose. And actually the very first operon that we looked at when we looked at the operon structure is actually the lac operon. And it's the lac operon that makes beta-galactosidase, the permease that we just talked about. It should be on this, yeah, there we go. So the permease here is LAC-Y, LAC-Z is the beta-galactosidase, and then there's another enzyme, we won't talk about that, acetylase, is LAC-A. So when lactose is present, the cell wants to make all of these. It wants to transcribe it and make the proteins, okay? And it does so by the RNA polymerase binding and making all of these components, okay? So if lactose is present, then that is what wants to happen. And so what happens if we'll start by looking at what happens if lactose is absent. Okay, so in the absence of lactose here, this is the normal scenario. In the absence of lactose, you have a repressor that's always bound to the operator. So when lactose is absent, here is the scenario. The repressor is bound and it's inhibiting synthesis of these genes. Okay, RNA polymerase can't access these genes, can't make the messenger RNA. What happens when lactose is present? Well, it's the opposite of that arginine situation. When the lactose is present, it acts as an inducer. It actually binds to the repressor, and actually you can see the change in shape of the repressor. The repressor can no longer bind to the operator. Okay, so lactose is acting as an inducer of the system. When it's present, the repressor can no longer bind. The RNA polymerase is free to transcribe these genes, and then they're translated into proteins. And these proteins are involved in the metabolism of lactose as an energy source. Okay, so this is the scenario when lactose is present. This is when lactose is absent. Okay, so it's quite an interesting and straightforward system. So again, just to keep us all awake, we've about just over 10 minutes. So when the question is, when the cell has lots of lactose, this sugar, what does it do? Does it activate the expression of lactose genes to help break down the sugar and use it? Or does it repress the expression of lactose genes when lactose is present? Okay, so think about that. And I can tell you the answer is 1. I'm glad to see that everybody has put the answer as well. Okay, so well done. We're still awake. I know there's a lot in this lecture, I realize that. So you're correct. The cell, if the cell is in the presence of lactose, so it needs to break it down. It needs to use it. Okay, and that's exactly it. So these questions are there to stimulate your understanding. So there's a slight complication. Is that when they were studying the system, and this shows you how even a bacteria can sense its environment so nicely. When they were studying the system, they started to study E. coli that's grown in a mixture of glucose and lactose. So what happens when E. coli is grown in a mixture of these sugars, glucose and lactose? Well, they found an interesting thing, that the E. coli would grow extremely well immediately. You can see the slope of this is quite high, so it's an extremely good growth. But it would utilize all the glucose in the system. So if you track the level of glucose, it goes down. The cell is utilizing the glucose for extremely good growth. And then there's this lag phase. The cell is getting ready for the next phase. And then you've got a slightly inferior growth. It's not quite as good. And this is when it's utilizing the lactose. Okay, and then the lactose goes down. So in other words, E. coli has a preference for utilizing glucose for efficient growth, and then it switches to lactose. Okay, so how does it do this? How does it sense, knowing what we know about the systems, how does it sense that and utilize all the glucose first, and then switch to lactose? It must have a system for regulating when the lac operon is expressed in the presence of glucose. So what happens when we add glucose to the system? Well, they did some very clever experiments, and they found a protein called CAP, which is called the catabolite activator protein, CAP. Again, this is in every you'll find this information online, or every microbiology textbook describes this. And here's that dioxic growth. And what they found was that gene expression is actually controlled by a small molecule called cyclic AMP. That's a little chemical in the cell. It's a tiny little chemical, cyclic AMP, which activates CAP, and it induces expression of genes required for lactose breakdown. Okay, and this is the key thing here in red. This is what you have to note. It's that cyclic AMP is low when glucose is present. Okay, so if you were to look at levels of cyclic AMP in the system, you'd see that actually they're extremely low here. So the cell has very low cyclic AMP, then it runs out of glucose, cyclic AMP increases, and the cell switches on the lactose genes. Okay, so what you really have to learn and remember is that cyclic AMP is low in the presence of glucose, but high in the presence of lactose. Okay, or when it's high when glucose is exhausted. So cyclic AMP goes up here, and that activates the catabolite activator protein, the activator protein. So this is the first case where we see activation. Okay, so we start to see the use of this the LAC-I site, the activator site. So in this scenario, cyclic AMP, when cyclic AMP is high, the activator is high, and you get this interaction between the activator and the RNA polymerase, which activates the expression of these genes. Okay, and it's actually better in the next slide. Okay, so cells use an activator called CAP, catabolite activator protein, which is actually stimulated by cyclic AMP. And this stimulated CAP increases the expression of the structural genes. It's actually better in this example because you'll see that CAP binds to CAMP, to the cyclic AMP. This is activated, and that is an activator interaction which promotes expression of the genes. And the key thing is that cyclic AMP, is that on this? Cyclic AMP is only high when the glucose has been used. Okay, so when the glucose has all been used up, this is what happens. And lactose is the lactose structural genes. Yeah, this is the key one. There's a bit of repetition as the story progresses. This is the key one that you have to remember, that in the presence of glucose, cyclic AMP is inhibited, the adenylate cyclase is inhibited, you get low levels of cyclic AMP. But imagine when the glucose is all used up, glucose goes down, then you get a buildup of cyclic AMP, it activates the CAP protein, and this activates the expression of all of these systems. Okay, so this is what they worked out when they looked at the LAC operon. This allows the preferential use of glucose, and then when glucose is utilized, cyclic AMP increases binding to CAP, and you get an increase in expression. Okay, just check the time. Still got some time. So just what I've done is I've just very simply put all these different scenarios in place in as simple a way as we can to look at the different scenarios that can exist for the LAC operon. So in the presence of low glucose, so glucose is key. Glucose is the main regulator of the systems. When glucose levels are low, cyclic AMP is produced. Cyclic AMP attaches to CAP, okay, and this helps RNA polymerase bind to the promoter, resulting in high levels of transcription. So that's low glucose. In high glucose, no cyclic AMP is made. CAP cannot bind to the DNA, so transcription occurs only at a very low level. You don't get very much present. So it's really inhibiting the LAC operon. So that's at high glucose. So what happens when we bring lactose into the equation? If glucose is present and lactose is absent, sorry about all the complications. Glucose is present, so the and lactose is absent, so the repressor is bound. Okay. Glucose is present, so you don't get CAMP, you don't get cyclic AMP, so you don't get activation, but you also get repression because lactose is absent. What about when glucose is present and lactose is present? Here you get very little expression. You get the catabolite activator protein is not active, so you get, because you have low cyclic AMP. So you don't get the activation. You do get some smaller level of expression, but it's not very much. Okay, because you don't get the activation. Okay, so this is from a textbook. Finally, the final bit on this section is that when glucose is absent and lactose is present, that's when you get the most activation, right at the end of that growth curve. You get cyclic AMP activates the process, and you get a lack of a repressor. The repressor is not active, and that way you've got optimal expression of the lac operon. Okay, so hopefully what you'll be able to do is go back over these slides. You'll be able to see, well, the way to study this is to draw it out for yourself and see what happens in the different scenarios. And I think it should make sense based on the way we presented it. And what it shows is that cells are very clever. They're very complicated. They're clever, and they can adapt to their environment properly. Okay. So just got two more slides, and this is really to bring in, why do we study this? You know, the scientists who worked this all out were very, very clever people. It's really an important part of the history of biology, but also it's telling us a lot about how cells react in different environments. So this is pretty advanced biochemistry of the cell. And all I wanted to do, you don't have to learn this slide. Please don't learn about I-kappa kinase and all this. We're not going to go through that. This slide is there to show you that the systems that are in place in bacteria, also a lot of the similar systems occur in eukaryotic cells. Okay, the point here is that eukaryotic cell can also respond to its environment. And actually, this is a thing called the NF-kappa B system, which you'll be familiar with maybe from immunology. In immunology, a T cell in the blood cell, white blood cell, it's obviously going to respond to its environment. So what happens in immunology is the T cell gets hit by all these cytokines. The cytokines switch on signals. The T cell responds by making new proteins, changing its shape, changing its receptor proteins. So all of these things are down to gene regulation. Okay, so how a T cell responds to an antigen or to a receptor or to a cytokine, this is all gene expression. Okay, and that's why, remember I showed you the picture of Mohamed Salah earlier on. You know, that's why he's so complicated. That's why we're all so complicated, because we've got all these signals coming into the cell, and they are influencing how genes are expressed. So it's important stuff. Oh, there's Mohamed. There, Salah. So, so in summary, this is I'll just read through these quickly. One more minute. Proteins are important for cell biology, but they must be expressed only when they're needed. They're subject to induction. Changes to the environment, heat shock, nutrients, antibiotics for bacteria, and maybe cytokines in the immune system for humans, they require that cells change their range of proteins in order to adapt. Cells regulate this at the level of transcription. That's the most important thing, transcriptional regulation. Cells therefore only make messenger RNA for the proteins that they need. And we have outlined how this transcriptional regulation occurs through activation of gene expression, repression of protein expression, two-component systems, and so on. Okay, so we've looked at some of the mechanisms. Examples are the arginine operon. We didn't cover the tryptophan operon. We don't need to. But the lactose operon and vancomycin resistance, these are all different things that the cell is responding to, which we showed in this lecture. Okay. Whew. There's a lot in that lecture. There's quite a lot in that lecture. I appreciate that. But it's fundamental biology. So here is why it's important in pharmacy. Increase our understanding of cell biology, use in genetic engineering. Need to understand molecular biology, so clone express genes. Remember when they cloned the insulin gene genes, they put it under the expression of beta-galactosidase. And we need to understand how pathogens adapt and can sense the environment. So these are fundamental concepts. And that is the last slide, I believe. So the slide before the summary slide. That would be this slide. So just very quickly, somebody's asked me to explain this. What I want you to do is I want you, sorry, take that off. I want you to ignore all of this sort of information. And all this slide is saying is that if you take a human cell or a mammalian cell, and this could be a T cell in the immune system, so a white blood cell in the immune system. A white blood cell is responding also to lots of different signals. Okay, so just to get away from bacteria, back into more complicated cells like a T cell, lots of signals are hitting that T cell. They could be different cytokines, different signals, could be an antigen that it's interacting with. And these signals are communicating to the cell to change the expression of lots of different proteins in the cell. Okay, so all of these are just communication pathways. Maybe we don't need all that detail. The important point is the signal induces a receptor, and that basically changes the gene expression profile. And at the level of transcription, you get different changed function in the cell. So what I'm saying here is that it's not just in bacteria, but also in human cells, they respond to their environment to make these changes. Okay. Okay, so I hope that answers everything. There's one more chat. Okay, thanks very much. So I will let you go. And next week you have Owen Fleming, who's in the Department of Biochemistry here. So Owen Fleming will be telling you a bit more about cloning and biotechnology, maybe using some of these concepts that we've covered, but more about the biotechnology industry and how we can use some of this information to make insulin and other proteins for biotech use. Okay. Okay. So you can leave now. We finished this lecture. Okay. So thank you all. Thank you. There will be a recording of the lecture. Yes. It is recording, yeah. I can see that it's been recorded. I'm not in control of the recording, but Dr. Marwa will make this available. Okay. So thank you, everybody. And I'll see you in 3 weeks' time, actually. You'll have 2 more lectures with Dr. Fleming, and then I will see you in the last lecture of our series. Okay. So I will stop sharing. And you can all go to your next lecture and have a nice evening. And I'll end this now, actually.